Today, let us have a discussion on a topic called diuretics. Today our topic is about diuretics. So, before going into detail of the term diuretics, a bright student should know the difference between two terms. That is, first one is natriuretics, while the second one is diuretics. A bright student should know the difference between natriuretics and diuretics. So, what are natriuretics? Natriuretics are the class of drugs. Whenever these are administered to the patient, then natri is nothing but natri means sodium. So, whenever these natriuretic drugs are given to the person, it increases the loss of sodium ions in the urine, are called natriuretic drugs. Natriuretic drugs are the drugs which will increases the loss of sodium in the urine of a patient, are called natriuretics. So, what are diuretics? Diuretics are the drugs which increases the outflow, that means which increases the quantity of the urine, that means it increases the volume of the urine which is excreting by a person within 1 day. So this is the difference between natriuretics and diuretics. Natriuretics means it will increases the sodium concentration within the urine of a person, whereas diuretics means it increases the volume of the urine which is excreting. So these are two important terms to be remembered by a bright student, that is natriuretics and diuretics. So natriuretics and diuretics. Now, before going into detail of diuretics, let us study normal physiology of nephron. That means without normal physiology of nephron, these diuretics is very much hard to understand. So now let us discuss about the normal physiology of nephron in short time. Normal physiology of nephron. Now let us consider, as our body is small, this is our cardiovascular system, this is our heart. Let us consider this is cardiovascular system and this is our heart. Inside it is consist of blood. So from cardiovascular system, the blood will be reaching to the kidneys. So this is Bowman's capsule. From the cardiovascular system, blood will be reaching towards this. This is efferent arteriole, and here it is a glomeruli. This is a glomeruli, and here this is the efferent arteriole. So this is the first part of nephron, which is Bowman's capsule. This is a glomerulus and this is a Bowman's capsule. Now continuing to this Bowman's capsule, this is called PCT. Now this PCT is lined by special type of epithelium. It is lined by special type of epithelium. So this is the next part. Let us consider this is one nephron and this is the blood circulation to the nephron. Normally, as one kidney consists of 1.2 million nephrons. So out of this is the only one nephron which I have taken here. Now this first part is called proximal convoluted tubule, whereas this is called descending loop of Henle, and here it is also made up of different types of cells, epithelial cells. It is also made up of different kinds of cells. And here this is the ascending loop of Henle. So within the ascending loop of Henle, it is divided into two regions. One is, this is thick ascending loop of Henle, this is thin ascending loop of Henle. This is thin ascending loop of Henle. Thin ascending loop of Henle, whereas this is thick ascending loop of Henle. It is also consists of specialized kind of epithelium. This is loop of Henle. Now next to the loop of Henle, that is special structure called, this is DCT, and here this DCT is also made up of special kind of cells. This is DCT, made up of special kinds of cells. All are made up of epithelial type of cells. And next to this DCT, next to that of DCT, next part is collecting duct. Now this collecting duct is also made, this is a collecting duct. It is also made up of different types of epithelial cells, and it is made up of different types of epithelial cells. So this is the complete structure of nephron. So our topic is normally how much in healthy individual, how much amount of the blood will be coming to the nephron per 1 minute. Once again, how much amount of the blood will be coming to the nephron in a healthy individual in 1 minute is, let us consider, it is dependent on cardiac output. So normally cardiac output is 5 liters per 1 minute. So the amount of the blood which is coming out from the ventricles per 1 minute is 5 liters. 5 liters per 1 minute. So cardiac output is 5 liters per 1 minute. Now out of this 5 liters of blood which is coming out from the heart, out of this 5 liters, that means 5000 ml, 5 liters means 5000 ml. Out of this 5000 ml, how much amount of the blood is reaching to the kidneys for the filtration? This is the question. Out of 5 liters of blood which is pumped out from the ventricles, how much amount of the blood will reach the nephron for the filtration process, or which is involved in urine formation process? Now out of these 5 liters, 20% is going to the nephron. Out of this 20%, only 20% of blood is going to that of nephron. Out of this 20% means only 1 liter. 1 liter per minute. Only 1 liter of blood is going to the nephrons, out of which it is pumped by cardiovascular system. Now once again, clearly, if 5 liters of blood is pumped out from the ventricle per 1 minute, out of that only 20% of the blood is reaching to that of nephron for filtration process. That 20% is nothing but 1 liter. So 1 liter means 1000 ml. 1000 ml. So 1000 ml of blood is reaching the nephron from the cardiovascular system for the filtration process and which is involved in urine formation process. So this is the information which is known by every student, but a bright student should remember that whatever the blood which is coming towards the nephrons having compositions of two compounds. So blood is made up of two components. One is cells while other one is plasma. Cells and as well as plasma. So in normal blood, cells are having percentage of 40%, whereas plasma concentration is 60%. Our blood is composed of cells as well as plasma. Out of that, 60% is composition of plasma, whereas 40% is composition of cells. So out of this, here 1000 ml of blood is entering into the nephron. So out of this, it is again 60% is plasma, 40% are cells. So totally 1000 ml of blood. 1000 ml of blood is reaching to the nephron. Out of this 1000 ml, 40% is the plasma. 60% is the plasma. 40% are the cells. So if 60% of plasma means out of this 1000 ml, only 600 ml is 600 ml of plasma is entering into the Bowman's capsule. So this is information to be known by bright student, that is out of this 1000 ml of blood which is entering into the nephron from the cardiovascular system, only the 600 ml of plasma is utilized for the urine formation and filtration. Out of this remaining 40% will be moving out. So here only 60%, that means plasma is used for the filtration process. So the how much amount of quantity is coming towards this nephron is only the 600 ml. Only the 600 ml is coming to the filtration process. Now out of this 600 ml, let us consider this is 600 ml of plasma is entering inside the nephron. Whatever the 600 ml of blood which is entering inside the nephron, this 600 ml is completely not utilized. Some amount of the plasma will be wasted and some amount of the plasma will be utilized. Out of this 600 ml, only 10% of plasma is utilized. Out of this 600 ml, only 10%. Only 10% of plasma is utilized. So within this 600 ml, the 10% is about only 120 ml. 120 ml of plasma is entering into the nephron per 1 minute. So this is the information should be known by a bright student, that is for 1 minute 120 ml of plasma is entering into the nephron, which is released from the cardiovascular system. So normal student will say that 1 liter of blood will be entering into the nephron for the filtration process and urine formation process. But the answer from the bright student should be 120 ml of plasma will be entered into the nephron, that is Bowman's capsule, for the filtration process as well as urine formation process. Now this is the information will be known by a bright student. Now out of all this, how much amount of the volume of urine should be released out from the normal person? So let us consider one graph. Let us consider this is one graph. So here let us consider two lines. The normal amount of the normal amount of the urine which is excreted from the healthy individual for 1 day is about 1.5 liters. 1.5 liters per day. This is normal. Whereas somewhat this 1.5 can be increased up to 3.5 liters per day. 3.5 liters per day. So these are normal conditions. The normal conditions is more than 1.5 liters and less than 3.5 liters per day. This is normal condition. So here this is normal condition. This is about normal condition. So if any person or any patient is excreting the urine less than 1.5 liters per day, it is a condition called oliguria. Oliguria. Oliguria is a condition in which the patient will excrete the urine less than 1.5 liters per day. That is, less amount of urine will be excreted from the person is called oliguria. Now, coming to the next one, if the same patient excrete more than 3.5 liters of urine per 1 day, is called polyuria. Polyuria. So these two are the abnormal conditions. The normal condition is between more than 1.5 liters per day and less than 3.5 liters per day. This is normal condition. If the same patient excrete less than 1.5 liters per day, is called oliguria. If same patient excrete more than 3.5 liters per day, is called polyuria. So this is the normal volume of the urine which is excreted from the healthy individual. Now this is some basic introduction about. So one more point, concentration urine formation includes of 3 steps. One is, first step is glomerular filtration. Here it takes, this is the first step, that is glomerular filtration. So as the glomerular filtration is taking place, it takes the important proteins like plasma and water and sodium and chloride and bicarbonates, etc. Like this, all the substance like constituents of plasma will be moving inside of this Bowman's capsule. So this is the first step. Glomerular filtration is the first step. So whatever the components which are secreted will be moving in this manner. It will be moving in this manner, and at last it will form a drop. This is called urine. So this is the first step, that is glomerular filtration. So out of this glomerular filtration, next step is, there are some elements which are required for our body. So like sodium, bicarbonate, chlorides are required for our body. So whatever the components which are required to our body, which are released out from this plasma, will be reabsorbed back. It will be reabsorbed back at this PCT. So this is the second condition, second step. First one is glomerular filtration. Second one is tubular reabsorption. That is, the important constituents like sodium and important constituents like chloride and bicarbonate, that will be reabsorbed back. This is the second step. First step is glomerular filtration. Second step is tubular reabsorption. And after the tubular reabsorption, the leftover content will be moving outside. So while the unwanted material is moving in this manner, now our body has some unwanted materials. So there are some unwanted materials like ammonium. Now the unwanted materials will be secreted into this these processes. Now this is the third step. The first step is glomerular filtration, second step is tubular reabsorption, third one is tubular secretion. So the unwanted materials will be secreted inside this material, that is nephron, so that it will get excreted up along with the urine. So here along with the urine, along with this all constituent, it forms urine here. This is the urine. This is a drop of urine which is formed. So this is the normal physiology of nephron which is involved in urine formation. So now our topic, this is the introduction. Without this introduction, diuretics cannot be understood. So now let us have a discussion on the reabsorption, where diuretics are applied at that reabsorption conditions. Now, with the continuation of this lecture, let us study the reabsorption of sodium at different parts of nephron. Like, first let us discuss about different reabsorption of sodium in PCT. Next, let us discuss about reabsorption of sodium in distal convoluted tubule. And next, let us discuss about the reabsorption of sodium ions in loop of Henle. And next, let us discuss about the reabsorption of sodium ions in collecting duct. Now, the motto of all the diuretics which are used is it will increases the loss of sodium ions within the urine and increases the volume of the urine which is excreting out from a body. Now let us discuss about reabsorption. First, as our body is small, let us study one by one. Reabsorption of sodium ions in PCT. Reabsorption of sodium ions in PCT. So, first let us discuss about the normal physiology which is taking place in the reabsorption of sodium ions in PCT. After the normal physiology which is taking place, now after this normal physiology, we will study the pharmacology which is involved at this level of PCT. So now let us consider this is one Bowman's capsule and it is made up of, here this is PCT, it is made up of different kinds of cells. Now let us elongate one cell. Let us elongate one cell. Let us consider this is a nephron and this is one cell of PCT. So as it is made up of different kinds of cells, I have taken one large cell from that of PCT. So now here this is the glomeruli. Out of this glomeruli, sodium is coming out down and bicarbonates are also coming down and chlorides are also coming down, whereas water is also coming down, which is entering inside to that of Bowman's capsule. Now from that of Bowman's capsule, these sodium molecules will be reaching towards this PCT. So this is, let us consider this is one cell and this special area, let us consider this is the area this is the area which is facing towards the lumen of the nephron. So this is the side which is facing towards the lumen. So this membrane is called luminal membrane. This membrane is called luminal membrane, which is bilayered, which is made up of lipid bilayer. So these two layers, outer layer, sorry, upper layer and lower layer, is called lateral layers. These two are called lateral layers. Or lateral membrane, and this outermost layer, which is present opposite to that of luminal membrane, is called ventral membrane. This is luminal membrane and these two are the lateral membranes, whereas this is called ventral membrane. So this is one cell. Below it consists of again same type of cells. So out of this one cell is enlarged. Now what is the mechanism which is taking place in the reabsorption of sodium in PCT? So this is the PCT. Now what happening means there is one special protein which is present on the surface of membrane, which is integral protein. Let us consider this is one protein. Let us consider this is one protein which is present on the surface of luminal membrane. So whatever the sodium which is coming toward downside of this PCT, it will come to this side. So the function of this enzyme is it will take sodium ions inside. It will take the sodium ions inside. So with exchange of this sodium ions, one important form to remember is it will excrete the protons. That means there will be influx of sodium ions and there is efflux of potassium, the efflux of protons. So the name of this enzyme we'll be discussing later. The function of this enzyme is it will grab the sodium ions which are coming towards this side and it will helps in influx of this sodium ions. That means the sodium ions will be entering inside these cell, epithelial cells. Whereas whenever one sodium molecule is entered inside the cell, one proton molecule will be removed outside of the cell. So this is the phenomenon. So it involves in exchange of sodium and proton. So the name of this special protein is called sodium hydrogen or sodium proton transporter. Transporter. This is the name of the special enzyme, that is sodium hydrogen transporter. That is H plus means proton, sodium proton transporter. The function of this sodium proton transporter is it helps in influx of sodium ions and efflux of protons. So this is about normal cell where sodium concentration will be increased, but a good student should remember that on surface of this lateral membranes as well as ventral membranes there is one special protein is present. So these special proteins are called sodium potassium ATPases. Same, sodium potassium ATPase. So this is sodium potassium ATPases, which is present on every cell of lateral membrane and ventral membrane. So the function of this sodium potassium ATPases is it helps in efflux of 3 sodium ions towards outside and 2 potassium ions towards inner side. So as the sodium ions are entering inside of this epithelial cell with the help of this sodium proton transporter, these sodium molecules will reach the sodium potassium ATPases. It will reach the sodium potassium ATPases. As a result, it will attach to the sodium potassium ATPases and there will be efflux of sodium ions. So these efflux of sodium is nothing but it will be reabsorbed back. So again from here sodium will be entered into the bloodstream. That means there will be increase of sodium concentration which is required for our body. So whatever the sodium which is coming out from this sodium potassium ATPases will enter into the interstitial area. From the interstitial area, it will be entered into the bloodstream. So this is a normal method where the sodium is entered inside and will be moving out with the help of sodium potassium ATPases. Now there is one more mechanism. Here these are the bicarbonates. So these bicarbonates will be coming in towards the downside, whereas HCO3. These bicarbonates cannot move inside the cell without easily. So these H plus and HCO3 will be reacted together, will result in the formation of H2CO3. H2CO3 will be formed. So H2CO3 will be formed. So on the surface, after formation of H2CO3, on the surface of these membrane, that is luminal membrane, there is special enzyme called carbonic anhydrase. This is a special enzyme called carbonic anhydrase. So with the help of this carbonic anhydrase, these H2CO3 will be converted into carbon dioxide as well as water. So after the conversion of this H2CO3 into carbon dioxide and water, these carbon dioxide and water can easily pass through this luminal membrane, water and carbon dioxide. So it will enter into the membranes inside the cell. So carbon dioxide and water inside the cell, again it will get fused up. It results in formation of H2CO3. Again inside the cell, again there is internally present of carbonic anhydrase which is present in the cytoplasm. So with the help of this carbonic anhydrase which is present in the cytoplasm, these H2CO3 will be cleaved up into H plus HCO3 minus. That means it results in formation of H plus and HCO3 minus. So in this manner, with the help of the carbonic anhydrase, now these H2CO3 will be cleaved into two compounds, one is H plus, that is proton, and HCO3, that is bicarbonate. So how these H plus are present in the cell means this is the process with the help of carbonic anhydrase. This carbonic anhydrase will take up the H2CO, carbon dioxide and water into the cells, and it will be mixed up it results in formation of H2CO3. Again, with the help of carbonic anhydrase, this H2CO3 will be cleaved up into two compounds. One is H plus and HCO3 minus. So these H plus will be accumulated within the cell. Now, what is the usage of this H plus means? Whenever these sodium ions are coming into the Bowman's capsule for the filtration, if one sodium molecule is entered inside, this one H plus molecule will be moved outside. So in this way, these are useful for the storage purpose. When one sodium molecule will be entered inside, then one proton molecule will be moved outside. So in this way, these are useful. So this is the normal mechanism where reabsorption of sodium will be taking place with the help of two enzymes. One is called this sodium proton transporter, while second one is carbonic anhydrase. Carbonic anhydrase. So these are the two important enzymes or two important proteins which are present on surface of this luminal membrane, involves in the reabsorption process, where this sodium proton transporter involves in transportation of one sodium molecule from outside to the low side. So why it moves from outside to the low side means, here the concentration of outside the cell, that is inside the lumen, the concentration of sodium is more, whereas inside the cell the concentration of sodium will be less when compared to the outside. So as a result, sodium will be moved from outside to the inner side. So if the sodium has to move from outside to inner side, the concentration within the cell must be lower than that of lumen. Then how the concentration within the cell will be maintained less than that of luminal means? With the help of the proteins called sodium potassium ATPases. Because of presence of this sodium potassium ATPases, the concentration of sodium ions within the cell is lesser when compared to that of lumen. So as the concentration of sodium ions within the cell is lower than that of lumen, when sodium ions are moving from the glomeruli to the Bowman's capsule, these will get reabsorbed here because of low concentration of sodium ions in the epithelial cells of PCT. From the cells of these epithelial cells of PCT, it will be moved with the help of sodium potassium ATPases. From the sodium potassium ATPases, sodium will be moved to the interstitial area. From the interstitial area, it will be reached to that of, again, once again here. So here, this is the sodium moving out. It reaches the interstitial area. From the interstitial area, again, it is going into the blood vessels. So inside the blood vessels, the sodium concentration will be increased. So in this way, reabsorption of sodium will be taking place at the site of PCT. Now this is a normal physiology which is taking place. So in case of diuretics which are used in these areas are, the main motto is to increase the sodium concentration within the urine. So here it is a urine. So if the main motto of the diuretics which are used in this site of PCT is to increase the concentration of sodium ions within the urine, that is, it increases the loss of sodium ions in the urine. So what is the pharmacology which is involved in this area means, there are some drugs which are administered in this area. So at which area these drugs are administered in this area means, so as we know that there are only two proteins. So one is sodium proton transporter and second one is carbonic anhydrase. So there is only two chances, but the main important type of the drugs which are administered in reabsorption of sodium in PCT is, there are some class of drugs which blocks the carbonic anhydrase. So there are some group of drugs which will block this carbonic anhydrase. Carbonic anhydrase will get blocked. So if carbonic anhydrase enzyme is get blocked, then these H2CO3 cannot be cleaved into carbon dioxide and H2O. If this carbon dioxide and H2O do not form, they cannot enter into the epithelial cells. If carbon dioxide and water is not entered into the cell, it cannot get assembled again. They do not form the H2CO3. If H2CO3 is do not formed, they cannot be cleaved into H plus and HCO3 minus. If H plus and HCO3 minus are not formed, the concentration of the protons within the cell will get decreased. So if the proton concentration within the cell will get decreased, then whatever the sodium which is coming from the lumen into the cell cannot be entered, because whenever the function of this, if this carbonic anhydrase enzyme is get inhibited, automatically it will inhibit the function of this sodium proton transporter. How it blocks means this H plus concentration will be decreased. So if the H plus concentration is decreased, the function of this is there is exchange between the sodium and H plus molecules. If the H plus molecules are decreased, whatever the sodium which is coming into this cannot exchange the protons. So if there is no exchange of H plus ions, this sodium cannot be entered inside. So whatever the sodium which is present in the lumen cannot be entered inside. So without entering into the lumen, these sodium ions, they fail to enter into the lumen. As a result, it will move in this manner and will increase the sodium concentration within the urine. Sodium concentration will get increased. These are called group of drugs called natriuretics, which increases the concentration of sodium ions within the urine. So what are the special group of drugs which are used here? Means carbonic anhydrase inhibitors. So the drugs which are used here are carbonic anhydrase inhibitors. So what are the drugs which, let us consider here. This is the drug. This is a special group of special drug which blocks the carbonic anhydrase. So the name of the drugs are, first drug is acetazolamide. This is a one drug. Next drug is brinzolamide. And next one is dorzolamide. These are the three group of drugs which are administered for the carbonic anhydrase, which are considered as the carbonic anhydrase inhibitors. It will inhibit the carbonic anhydrase enzyme, then there is no production of H plus ions. If there is no production of H plus ions, then there is no influx of sodium ions, there is no reabsorption of sodium ions. So the reabsorption of sodium will get decreased. So here the reabsorption of sodium in the PCT will get decreased. So this is the first part of diuretics which are involved in PCT. Now next let us discuss about next part. This is about carbonic anhydrase inhibitors, which inhibits the enzyme called carbonic anhydrase. Now next let us discuss about reabsorption of sodium ions in thick ascending loop of Henle. reabsorption of sodium ions in TAL means thick ascending loop of Henle. So let us consider this is the second part. First part is about PCT and this is second part, that is thick ascending loop of Henle. This is a thick ascending loop of Henle. So it is made up of different kinds of cells. Now let us enlarge it some more bigger. Here this is the thick ascending loop of Henle. This is a PCT and descending loop of Henle. This is a descending loop of Henle, and here this is a thick ascending loop of, this is a thin ascending loop of Henle, and this is a thick ascending loop of Henle, and here it continues to the DCT and collecting duct. Now this thick ascending loop of Henle is made up of special type of epithelial cells. Now let us take one big epithelial cell. Let us consider this is one epithelial cell, which are similar type of cells are present at the sides. Now as we know that this layer is called luminal membrane, and here this layer is called ventral membrane. This is called ventral membrane, and these two are called lateral membranes. Luminal membrane, ventral membrane, and two lateral membranes. So we are talking about the concept of reabsorption of sodium ions in thick ascending loop of Henle. This is a thick ascending loop of Henle. So before going into that complete discussion, let us consider this is one nephron. So out of this nephron there are some regions where the water is permeable whereas water is impermeable. So whatever the plasma constant which are entering inside, so this is the region, where all the water is, this is the region where the water will be permeable. So in this area, from the Bowman's capsule, PCT and descending loop of Henle, up to the descending loop of Henle, the water will be permeable. So water reabsorption can be taking place in the Bowman's capsule, PCT and descending loop of Henle. So from the ascending loop of Henle, from this region to this region, it is completely impermeable to the water, whereas these regions from descending loop of Henle, that is thin descending loop of Henle and thick ascending loop of Henle, it is completely impermeable to the water. So this is the permeability and impermeability. Now coming further to the top, if coming to this DCT and collecting duct, so here in this area the permeability of water is mainly dependent on special hormone or special protein called ADH, that is vasopressin. If the vasopressin or ADH is hormone is present, these area will become permeable to that of water. If ADH is present, these whatever the fluid which is present inside these collecting ducts, at the region of collecting ducts, will be acting as permeable to that of water. If in case there is absence of ADH hormone, these region will act as impermeable to that of water. This is to be remembered by a bright student. So coming back to that of here, so here the sodium will be coming inside. In this manner, sodium will be coming towards the thick ascending loop of Henle. So here the sodium will be reaching. Large amount of sodium will be present. So on the surface of luminal membrane, there is special enzyme is present. So special protein is present. Name of that special protein we will be discussing later, but let us first discuss about the mechanism of this protein. So this is a special protein which is present on surface of this luminal membrane of cells of this thick ascending loop of Henle. So this is the special protein present on the, which is integral protein which is present on the this luminal membrane. Now what is the function of that special enzyme is, let us consider these are the sodium ions which are moving towards this area. Sodium ions are reached to this protein. Now with the help of this protein, sodium ions will be entering inside. Sodium ions will be entering towards inside. So along with that of sodium ions, it will take negatively charged chloride molecules. So before talking about negatively charged chloride molecules, along with that of sodium, it will take potassium ions also. It will take the potassium ions. So these are potassium ions. It will take potassium ions. So along with the sodium and potassium ions, they have capacity to take 2 chloride molecules. It will take 2 chloride molecules. Once again, this is a special protein which is present on surface of luminal membrane of epithelial cells of thick ascending loop of Henle. So the function of this special protein is, it will helps in reabsorption of sodium. That means sodium, whatever the sodium which is entered here will be moving inside of this cell which is present in the loop of Henle. That means sodium will be entered inside the cell with the help of this special enzyme. So while moving of the sodium ions, it will take potassium ions along with that. So it will take the potassium ions. So along with these 2 molecules, along with that of 2 molecules, it will take 2 chloride molecules. That means sodium, potassium and 2 chloride molecules will be moving towards outside. So the name of that enzyme is, as it is taking the sodium, potassium and chloride. So the name of that enzyme is sodium, potassium, 2-chloride cotransporter. This is the enzyme which plays key role in the reabsorption of sodium in thick ascending loop of Henle. So this is the enzyme called sodium potassium 2 chloride 2 chloro co-transporters. Sodium potassium 2 chloride co-transporters. It will take sodium, potassium and chloride. So as sodium will be taking, as we know that on every cell these are the sodium potassium ATPases. These are the sodium potassium ATPases. So whatever the sodium which is, let us consider here, this is also sodium potassium ATPases. So like sodium potassium ATPase. Now this sodium which is present will be moving to the, this is the sodium potassium ATPases, and will be moving to the interstitial area. From the interstitial area again it will reach to that of bloodstream. It will reach to the bloodstream. That means in this way the sodium will be reabsorbed back with the help of sodium potassium 2 chloride co-transporter. So the sodium which is coming in from that of lumen side into the cell will be balanced with that of sodium potassium ATPases. So whatever the sodium which is present inside the cell, which is came out from the lumen, will be balancing by going out with the sodium potassium ATPases. But the main problem is the concentration of potassium ions within the cell. As the potassium are moving towards the inside, as the concentration of potassium ions are moving inside the cell, then the potassium concentration within the cell will get increased. So potassium concentration within the cell is getting increased. So in order to overcome that, increase the concentration of potassium ions, our nature has beautifully designed that within the membrane of these lateral sides and ventral sides, there is a special protein or special protein which is present on a membrane called, this is a special protein that is called potassium deloader. This is a special protein called potassium deloader. So whatever the potassium which is present will be moving out. It will be moving out and it will be taking out. So it will be taking out. So in this way the potassium ions are also balanced. So if this is the normal physiology which is taking place, that is reabsorption of sodium ions in thick ascending loop of Henle. That means with the help of enzyme called sodium potassium 2 chloride co-transporter. This is the enzyme sodium potassium 2 chloride co-transporter. Now what is the pharmacology which is involved means, there is special type of pharmacology which are called as loop diuretics. Special drugs which are called loop diuretics. So loop diuretics which are acting at the region of loop of Henle. So there are some special class of drugs which will block these sodium potassium 2 chloride co-transporter. It will get blocked. So because of blockage of this sodium potassium 2 chloro co-transporter, the sodium ions cannot be enter inside. So sodium ions cannot get enter inside the cell. If the sodium ions cannot get enter inside the cell, this sodium cannot go out from the sodium potassium ATPases. So if the sodium concentration within the cell will get decreased, the reabsorption of the sodium will be decreases because of blockage of this sodium potassium 2 chloro cotransporters. So the first thing is it will block the sodium potassium 2 chloro cotransporter, so that the sodium cannot enter inside the cell because of blockage. So excess amount of sodium will be accumulated within the loop of Henle. Next one, because of blockage of this sodium potassium 2 chloro cotransporter, the potassium concentration within the loop will get increased. The potassium concentration will get also increased. So as a result, the potassium concentration and sodium concentration will get increased. And one more important point to remember is, as the solute is moving from fluid is moving in this manner, there is secretion of calcium and magnesium ions. There is secretion of calcium and magnesium ions. So if this enzyme, that is sodium potassium 2 chloro cotransporter enzyme, is blocked, the sodium concentration within the loop will get increased. So already the sodium are positively charged, so the electropositivity within the loop of Henle will get increased. So as the electropositivity within the loop of Henle will get increased, these calcium and magnesium are also positively charged. So positively charged will get repelled with positively charged. So as a result, these calcium and magnesium will be reabsorbed back. Because of blockage of this sodium potassium 2 chloro cotransporter, there is a positive response that it will helps in reabsorption of calcium and magnesium ions, because these are coming along with that of sodium in this fluid. So calcium and magnesium will get reabsorbed. So if this enzyme is blocked, this potassium concentration will get increased. So as a result, first condition is there is increase in sodium concentration, that is natriuremia. First side effect is, let us discuss about if excessive amount of before discussing about side effects, we have discussed about the mechanism of action of drugs which are blocking this area, that it blocks this enzyme sodium potassium 2 chloro cotransporter. That means there is no influx of sodium and there is no efflux of sodium from the sodium potassium ATPases. There is no reabsorption. As there is no reabsorption, the sodium will be moving in this manner. Sodium and other solutes will be moving in this manner and will be reaching out in the urine. So there is loss of sodium in that of urine. So that condition is called natriuremia. So what are the group of drugs which comes under the sodium potassium 2 chloro cotransporters? Means furosemide. First drug is furosemide. First drug is furosemide, and next one is bumetadine. Bumetanide. Bumetanide, bumetanide, furosemide and bumetanide. So these are the two drugs. There are some other drugs also, but at least two drugs are important to remember. That is, furosemide and bumetanide are the two drugs which blocks the sodium potassium 2 chloro cotransporter. So if there is blockage of this enzyme, there is no influx of sodium and there is no reabsorption of sodium. So if any person is taking the furosemide and bumetanide for longer period of time, there is increase of concentration of sodium ions in the urine. So that condition is called natriuremia. This is the first side effect. First side effect of natriuremia, excessive usage of furosemide. Next one is, if there is the function of this furosemide is it blocks the sodium potassium 2 chloro cotransporter. So if it gets blocked, the potassium within the cell, that is potassium concentration is also increased. That means the potassium concentration in the loop of Henle will get increased, and ultimately it will reach the collecting duct and it is present in the urine. So the presence of potassium in the urine is called Kellyuria. That is second one, Kellyuria. Kellyurasis or Kellyuria, antiurasis and antiuria. And there is also presence of calcium ions. So if excessive usage of furosemide is done, if a person is taking furosemide for longer period, there is also a side effect called calciuria. This is natriuria, Kellyuria and calciuria. These are three side effects for the excessive use, for long-term usage of this furosemide. So these are some drugs which are considered as loop diuretics, which are administered at the region of loop of Henle, which blocks the sodium potassium 2-chloro cotransporters. So this is the second part of nephron, that is reabsorption of drugs which are used in reabsorption of sodium in thick ascending loop of Henle. Now let us consider next topic, that is reabsorption of sodium in DCT. Reabsorption of sodium in DCT. So let us consider this is this PCT loop of Henle and ascending loop of Henle, and here this is the DCT. This is the DCT, and it is made up of different types of cells, different types of epithelial cells. So out of that, let us draw one big cell here. This is one big cell of DCT. So we already know that this is the luminal membrane, which is double layered, and this is the ventral membrane, and these are the lateral membranes. Lateral membranes, luminal membrane, ventral membrane. Now, so already we know that here there is presence of sodium potassium ATPases. These are the sodium potassium ATPases. Sodium potassium ATPases. So as sodium ions are moving in this area, sodium ions are moving in this area. So it must be reabsorbed back. In this, how reabsorption will be taking place means, let us consider this is one enzyme which is present on surface of luminal membrane. Here sodium is coming. So there is an enzyme or protein which is present on the integral part of luminal membrane. So before knowing the name of that enzyme, let us study the function of that enzyme. This function of that enzyme is, whatever the sodium which is coming towards the DCT, it will grab the sodium ions towards the inside. It will grab the sodium ions towards the inner side. So along with that of sodium ions, it will also bring one more negatively charged molecule. That means it brings the chloride molecules. It will bring the chloride molecules. So as it is bringing sodium ions and chloride ions, the name of that enzyme is sodium chloride transporter. Sodium chloride transporter. This is a special enzyme which plays key role in the reabsorption of sodium ions in the DCT. So the name of this enzyme is sodium chloride co-transporter. So it takes the sodium ions and chloride ions inside. Whatever the sodium ions which are present will reach the sodium potassium ATPases. As a result, there will be efflux of sodium ions. So from this efflux of sodium ions, it will reach the interstitial area. From the interstitial area, again, it will reach the bloodstream. So as a result, it will get the sodium concentration within the blood will get increased. So sodium is reabsorbed. What about the chloride? What are the chloride molecules which are present? Again, there are special channels are present on ventral membrane. So the special channels which are present on ventral membrane are called chloride channels. So these chloride channels will get opened up and these chloride ions will be moving out. So these chloride ions which are moving out will again enter into the bloodstream. It will enter into the bloodstream. So this is a normal physiology which is taking place. But in some conditions, the drugs which are administered, what is the pharmacology which is involved in this area is, the drugs which are administered in this area are called thiazides or thiazides like diuretics. So the drugs which are administered in DCT are called thiazides or else thiazide like diuretics. So what is the pharmacology means? The thiazides. Different types of drugs are used like thiazides. Thiazides are used. So what is the function of thiazides? These, whatever the thiazides are present, it will block the sodium chloride transporter. It will block the sodium chloride transporter. That means thiazides will block the sodium chloride transporter. These are thiazides. It will sit on this sodium chloride co-transporter. Sodium chloride transporter, and it will block the sodium chloride transporter. So if this sodium chloride transporter is get blocked, then whatever the sodium which is moving out in this manner cannot be entered into the lumen of the cell. So if there is no entry of sodium ions into the lumen of the cell, there is no efflux of sodium from that of sodium potassium ATPases. If there is no efflux of sodium from the sodium potassium ATPases, then there is no reabsorption of sodium into the bloodstream. So as a result, the blood concentration of sodium within the bloodstream will get decreased because of decrease of reabsorption of sodium. So as a result, if the sodium fails to reabsorb, it will move in this manner. it will move in this manner and present in the urine. So it will increases the concentration of urine, so concentration of sodium within the urine, and increases the concentration and increases the volume of the urine. So in this way, thiazides are used for blocking the sodium chloride co-transporters. So what are the drugs which comes under thiazides are? Let us consider first drug, that is chlorothiazides. First drug or second one is hydrochlorothiazide. There are many drugs, but at least we should remember two drugs. That is, first one is chlorothiazide, second one hydrochlorothiazide. Hydrochlorothiazide and chlorothiazide. So these are the drugs which are administered in the inhibiting the reabsorption of sodium in the DCT. That means it increases the concentration of sodium ions within the urine. So this is the third type of drugs which are used as diuretics. Now coming to the last part, that is now coming back to the last part, that is reabsorption of sodium in collecting tubules. Reabsorption of sodium in collecting tubules. Now let us consider this is this is one nephron, and this is a PCT, Bowman's capsule, PCT, loop of Henle, and DCT, and here these are the collecting ducts. So it is also made up of different types of epithelial cells. Let us enlarge one cell here. This is one cell, and we also know that this is luminal membrane, this is ventral membrane, and these are the lateral membranes. Luminal membrane, ventral membrane, and lateral membranes. Now, as we know that the sodium is moving in this manner, and water bicarbonates are moving in this manner. Sodium has failed at the region of, it has failed the reabsorption at the region of PCT, and at the region of loop of Henle, and at the region of DCT. It reaches the collecting duct. Now, what are the functions, how the reabsorption will be taking place means, now on the surface of luminal membrane there are special channels are present. These are the special channels. So these are the special channels which are present on surface of luminal membrane. These special channels are called sodium channels. So these are whatever the channels which are, whatever the receptor proteins which are present at the PCT, loop of Henle, DCT are not the channels, but these are the sodium channels. But these are not voltage gated channels. These are specialized channels which are present at the only collecting ducts, collecting tubules or collecting tubules. Now these are called sodium channels. These are called the sodium channels. So whatever the sodium which is entering at this collecting duct will reach the sodium channels. From these sodium channels, it will enter inside the cytoplasm of this epithelial cell of collecting tubule. Now, as we know that there are sodium potassium ATPases. These are the sodium potassium ATPases. So whatever the sodium which is present inside the cell, which is entered because of this help of these sodium channels, will be moving in this area. There will be efflux of sodium ions. So one point to remember is sodium potassium ATPases is, it will take 3 sodium ions outside and it will bring 2 potassium ions inside. So now this is the matter or information which will be known by every student, but an intelligent student should remember the important point that if the sodium concentration in this fluid is increasing or it fails to reabsorb at these conditions or at these areas, the sodium levels will reach the collecting duct. So if the more concentration of sodium is present in this fluid which is reaching the collecting duct, so the sodium will be taken up by the sodium channels. So as a result, when the sodium channels are taking up the sodium ions, these sodium channels will take the sodium ions. As a result, whatever the sodium which is accumulated within the cell will be taken up by the sodium potassium ATPases. So by the help of sodium potassium ATPases, 3 sodium ions will be moving out and 2 potassium ions will be moving inside. So if the sodium concentration is more, the sodium influx will be more. If there is more influx, sodium influx is more, sodium concentration within the cell will be increased more. If the sodium concentration within the cell increases more, as a result, the work on the sodium potassium ATPases will be increased. So if the work on sodium potassium ATPases is increased, there will be more efflux of sodium ions. If there is more efflux of sodium ions, there is more influx of potassium ions. If there is more influx of potassium ions, the potassium concentration will be increased. There will be increase of potassium ions. So there will be increase in the concentration of potassium ions. So the concentration of potassium ions within the cell will get increased. As a result, there are special channels are present. So these special channels are called voltage sensitive potassium channels. So as the potassium concentration within the cell is increased, electropositivity within the cell will get increased. So as the electronegativity becomes electropositivity, the positive nature within the cell will get increased. Because of increase of electropositivity, these potassium ions will get opened up. So as the potassium ions are opened up, potassium will be moved outside. Once again, if there is more sodium concentration is more, sodium influx will be more. If sodium influx is more with the help of sodium channels, so work on sodium potassium ATPases will be more, sodium efflux will be more, and potassium influx will be more. As the potassium influx is more, potassium concentration within the cell is increased more. If there is electropositivity increases, then the work on the potassium diureters will get increased. So potassium diureters will get activated, that is potassium voltage sensitive potassium channels will get activated. As a result, the potassium ions will be moving outside. So as a result, urine will be formed. So whatever the urine which is formed will consist of sodium ions as well as potassium ions. So it is condition called cheliuresis. ketolysis condition can be seen. So in this way, sodium and potassium will be present in the urine drop. As a result, this is the normal physiology by reabsorption. So here some whatever the sodium which is present will be taken up by the sodium potassium ATPases. So from the sodium potassium ATPases, it will be going to that of bloodstream. Sodium will be enter into the bloodstream. Sodium will be enter into the bloodstream. As a result, the sodium will get reabsorbed back. So in this way, this is the normal physiology. But what is the pharmacology which is involved in this area means, if we need to increase the sodium concentration in case of dehydration or any condition, if the sodium concentration within the urine must be increased. So what are the conditions that can increase the sodium conditions means, there are two conditions. That now coming to the pharmacology, there are first type of pharmacology which is involved is, there are some special group of drugs which are working at these sodium channels. So these are special group of drugs which work on the sodium channels and will block the sodium channels. So whenever the sodium channels are blocked, the extra amount of sodium concentration which is present in the this fluid cannot activate the sodium channels. If these sodium channels do not activated, they cannot enter inside the cell. If the sodium concentration is less inside the cell, sodium potassium ATPases cannot get activated. If sodium potassium ATPases will not get activated, there is no efflux of sodium. If there is no efflux of sodium, this sodium cannot enter into the bloodstream. That means there is no reabsorption of sodium. If there is no reabsorption of the sodium content will be left over in the fluid only. That means sodium concentration will be left over in the fluid only, and it will be present in the urine. So these are the special group of drugs which block the sodium channels and will inhibit the sodium reabsorption. So first group of drugs are called sodium, that is epithelial. In our book it is written as epithelial cell sodium channel blockers, EANC blockers. Epithelial cell sodium channel blocker. Now what are the drugs which comes under these sodium channel blockers are amiloride, AMILORID. Amiloride is the first drug which work on the sodium channel and will inhibit the sodium channel. And next one is triamethene, TRIAMETHENE. Triamethene, 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 amiloride and triamethene. So these are the two drugs which work on the sodium potassium ATPases and will work on the sodium channels and inhibit the function of the sodium channels. So these are the special group of drugs. And one more class of drugs are present. As we know that these, what are the cells which are present, so they are called processing cells or principal cells which are present in the collecting duct, are associated with the action of aldosterone. Let us consider here adrenal cortex and adrenal medulla, and this is zona glomerulosa. From this zona glomerulosa, aldosterone will get activated. So this aldosterone which is activated will activate the principal cells. As a result of activation of these cells, aldosterone will enter into the cells. So after entering into the aldosterone, enter into the cells, it will activate the genetic material. So it will activate the genetic material and it will undergoes transcription and translation process. After the transcription and translation process, they form the special protein called aldosterone induced proteins. Aldosterone AIP called aldosterone induced proteins. So the function of that aldosterone induced proteins are, it will help in increase aldosterone induced proteins are, it will open the sodium channels. Aldosterone induced proteins will open the sodium channels if there is opening of sodium channels, there will be influx of sodium ions. If there is influx of sodium ions, there will be more activation of sodium potassium ATPases. That means there will be more reabsorption of sodium ions, and that is the first function. Second function is it will increases the sodium channels on the lateral side, lateral membranes and ventral membranes, and these it will increases the concentration of sodium channels only on the luminal membrane, so that if there is sodium, if the number of sodium channels are increased on these luminal membrane, there will be more rapid reabsorption of sodium ions. So reabsorption will be increased. So in order to decrease the sodium reabsorption, there are some group of drugs are present which will block the aldosterone receptors. So there are some second group of drugs are called aldosterone antagonist. So the special drug is called spironolactone. Spironolactone and eplerenone. These are the two group of drugs which will block the this aldosterone receptors, that are spironolactone and eplerenone. So these, we need to know one important term, that is potassium wasters. These potassium wasters. So these, what are the drugs which are used in collecting tubule, that is sodium channel blockers, and these aldosterone receptor antagonists are considered as potassium wasters. So it will increases the potassium levels. It will increases the levels of potassium within the urine. That means there will be loss of potassium. This is the information about diuretics. And last one is, that is osmotic diuretics. Osmotic diuretics are nothing but example for the osmotic diuretics are mannitol. So in at first let us consider, this is the nephron and mannitol will be given. At first mannitol will be given, but in ancient times, where the knowledge on drugs is not much, at that conditions, when mannitol is given, there is no usage of mannitol, and the mannitol is present, there is some concentration of mannitol is present, but after some discoveries and inventions, these, whatever the mannitol which is present will block the adrenergic receptors. It will block the adrenergic. Soon after they came to know that this, whatever the mannitol which is giving, will block the adrenergic receptors. So whenever the adrenergic receptors are blocked, the sodium cannot enter inside the cell. If there is sodium, no entry of sodium inside the cell, there will be no reabsorption of the sodium. If there is no reabsorption of the sodium, ultimately the sodium concentration within the urine will get increased, and ultimately there will be increase in the concentration of urine. So this is the information about diuretics.